Okay. Well, thanks everyone for joining us today. So it's my pleasure to introduce Bjorn. He's a professor in the Department of Mathematics at Brown University. His research covers a broad spectrum of topics in applied dynamical systems, computational and mathematical biology, and data science. So today he will tell us specifically more about how to use techniques from data science to link genotype and phenotype. Okay, so Bjorn, feel free to get started.
All right, thank you so much. So let me share my screen. All right, so this is, let me just rearrange my things slightly. Yeah, and so please feel free to interrupt me at any time. If you have any questions, just interrupt, and I'm happy to answer or try to answer them. So what I'm, whoops, going back. So I would like to talk about 3 different topics, and so basically spending about 10 to 15 minutes on each of them, that all have as the ultimate goal to think about the connection between genotype and phenotypes. And I should make a disclaimer at the beginning. So I'm not really a mathematical or computational, so I'm not really working on mathematical computational biology. I've sort of recently ventured into these areas, but really most of the work that I've been doing sort of via my graduate students, who I will acknowledge at the end, and then collaborators in computational biology labs. So there will be, sort of, as I said, 3 different topics. I want to talk a bit about zebrafish and understanding gene functionality in zebrafish, and then switching to understanding common gene structures that underpin groups of diseases. And then the final topic that I'm going to talk about that's more recent is thinking about aligning single cell data. So there's been, over the past 5 years or so, maybe a bit longer, people have been extremely successful at doing experiments on single cells and using various different techniques to extract, for instance, gene expression data, methylation data, and the problem with that is that you have many different cells from which you extract these data. And what you would like to do, hopefully eventually, is to compare these data between potentially different cells and different modalities, different types of experiments. And so I'm going to talk a little bit about one technique that we looked into that helps with that. So the first thing I would like to talk about is zebrafish. And so these are small fish, a few centimeters long. And one of the distinguishing criteria, feature that they have is that they have these stripes on their main body. They're formed by pigment cells. They're self-organized. There is, depending on how you count, 3 types of pigment cells, but 2 of them can change form and come up in different forms. And during the development of these zebrafish, these stripes form spontaneously and then begin to grow in this case in the vertical direction. So you grow them stripe by stripe slowly as the fish grows. There's a large number of genes involved in regulating this. And what biologists have been trying to understand is really what underpins, what are the genetic structures, the mechanism, the functionalities that they enforce that enables these stripes to grow. I'm going to show you pictures on the next slide. There's also a number of viable mutants that produce very different patterns on the skin. And you can use these experiments and these mutants to try to understand which genes are potentially knocked out by these mutants and how that affects stripe formation. And so the close-up that actually, sorry, I forgot to do this. Let me just go back and do my annotation scheme so that I have my pointer. Here we go. So what you see here is a close-up of that area of that fish, and so you really see the individual pigment cells. So you see the black ones, the melanophores. They're difficult to see here, but you see orange ones over here. They're a little bit more clearly to see here. And then there's also these silvery cells, bluish cells that come up here, which are called iridophores. So the black cells are often called melanophores. The yellowish cells are xanthophores, and then these bluish silvery cells are iridophores. And so these are sort of 2 different illustrations of these individual pigment cells. And so the questions is, as I said, is connect sort of gene expressions, gene functionality, and then the phenotypes that you observe. I showed 2 different fish here. One is Danio, which is the main focus of what people do experiments on, but there's a whole family of Danio fish, and so Margaritatus, for instance, featured spots instead of stripes. And so our goal was to use Asian-based models to test hypotheses and try to gain insight into what knocking out specific genes, what that impact might be on the patterns that you observe. And what I'm going to talk about today is a way of trying to quantify patterns that emerge either from these Asian-based models or from experimental images that biologists take. So in sort of earlier work that Alexandria Volkaning, one of my former students, and I have done, we developed an Asian-based model that describes stripe formation on zebrafish. And really, when we try to guess or estimate the parameters and try to fit the model to experiments, we really did this by hand. We really looked at, Alexandria basically ran simulations, looked at the outputs, measured, so try to understand, for instance, typical cell-to-cell distances, width of stripe patterns, number of stripe patterns, and try to match that. And the goal of what I'm going to describe is really how can we automate that and get quantify pattern features across scales, from cell-to-cell distances to macroscopic features like stripe alignment, stripe width, and then ideally do the same thing for experimental images. And so here's just 3 of the mutants that people have looked at. These are early stage mutations that are essentially characterized by missing pigment cell types. So each of these different mutations, shady, nacro, and pfeffer, lacks one of these 3 cell types that I outlined before. And then we see that the stripe patterns are no longer present. You either see these labyrinthic type patterns for shady, and then you see spots for pfeffer and for nacro. And you still see some kind of directionality. They're still sort of aligned somewhat along stripes, but the stripe pattern by itself is broken up. So the first thing I would like to talk briefly about is the Asian-based model that we used, and that we then would like to combine with an algorithm that can actually quantify the output of this model. And so there's sort of a number of different things one has to take into account. The first one is that these cells move. They move slowly, but they can move. They basically signal to each other and then either typically repel each other, so move away from other cells. And so we model this by a differential equation for the cell position. So we think of these cells as idealized point masses that move around. And then we have laws that govern, depending on typically near cell-to-cell signaling, how they would move. The next thing that can happen is they can be born, they can die, and they can also differentiate. In addition, 2 of these cell types, namely xanthophores and iridophores, can change form. And we're involving this by having random discrete time events where we check for these events and then sort of incorporate them into the model. So this means that every couple of hours, in terms of time that we model, we just basically check whether a cell should die, whether a new cell should be born at a certain position, or whether a cell wants to differentiate or change type. And then we adjust the number of ODEs, because that depends on what cells we have, and then again run for a few hours and then go back and forth between these models.
So just a question: the cell position is not a diffusion, right? It's just deterministic.
Correct. It's not a diffusion. So it's deterministic. And so there's essentially 3 different length scales over which these cells see each other. It's really chemical signaling. They have dendrites and pseudopodia that they reach out with and sense each other. And we're basically taking this into account by looking at nearest neighbors over these different scales and then computing a resulting force. If you add diffusion to the model, which we have done, it doesn't change the results in any way. So they would just have like this additional diffusive behavior, random walk model behavior in there, but it wouldn't change the overall dynamics of the outcomes and of the patterns that you see.
Thank you.
Sure. Whoops, just focus here again. Okay, and so what we want to do is we want to sort of take outputs like these 2 pictures, one from wild type and one from pfeffer, and we would like to quantify them. And so here's sort of the type of features that we would like to capture. We would like to compute average cell-to-cell distances within cell types and also across cell types. We want to count the number of stripes and the number of spots. We want to think about sort of stripe width and also the straightness, how straight or how curvy these stripes are. For spots, we want to see how large they are in terms of number of cells involved, and also how round they are and whether they are aligned along stripes or not. And then we would like to capture time-dependent events, like when do new stripes form, when do these cells, these spots form, and when does their number change. And so ideally, this should be automated, it should be data-driven, it should be suitable for both agent-based models, like the one I just described, as well as for data extracted from images, and it should work across scales. So even though we have some information about typical cell-to-cell distances and just the typical length scales, we don't know for sure. I mean, they are variable. They can vary from model output to model output and from fish to fish. And so ideally, the approach would account for that and not require like explicitly set length scales for it to work. And so what we decided on using was a combination of topological data analysis and clustering. And I would like to describe a little bit more of what the topological data analysis, the TDA part is. And the clustering is a pretty straightforward clustering methods that we use. And that's primarily for the spots and the number of cells within spots. So let me talk a little bit about TDA. And I'm not sure how many of you have seen this, but just one slide on just how that approach works. So imagine that these are your data. And so one guess that you might have is that these data come from sampling from a figure 8. Right, so it looks like approximately a figure 8. And so the question is, because you have single data points, how do you can gain some confidence or some information about whether that really is sampled from a figure 8 or not, when you cannot sample more points? So you just have those points available. How would you quantify that? And so the idea is that you enlarge your data points. So instead of thinking of them as points, you think about them as little balls, and the radius of the ball can change. And what we're doing is we're going to pick this radius initially 0. So we just have our single data points. And then I'm going to continuously increase the radius of that ball, look at the union of all of these balls, and try to understand what the structure of the data then looks like. So for instance, if I'm enlarging to a certain radius and I pick the radius to be just indicated by these balls, you see that starting with the initial figure 8, I really have a large number of connected components. Every single data point is its own connected component. And I really don't see any structure unless I start connecting these data points somewhere by lines or somewhere else. But once I increase the size of the radius, I actually see structures emerging. So for instance, here now I see just a single connected component. I can go from any data point through another data point through that union of balls centered at these data points. And I also see that I see sort of this first circle here that I think should be a part of the figure 8 over here. And if I keep increasing, then eventually I have 2 of these circles. So I really see sort of something that resembles a figure 8. These 2 data points here now join. And if I would continue doing this, at some point I would just see a huge ball. Everything would be filled up with these union of balls of these centered at these different data points. And so the idea is now that I can use the scale R that I can change from 0 to a larger value and see what the changes in the number of circles, the number of connected components are. So if I really change that change this entire set of radii, I can use sort of a diagram like shown here on the right to quantify when certain features emerge and when they disappear. So for each radius, I'm going to check the number of connected components. So you see in the lower bottom, and then basically I check when do they disappear. So what I see is for very small radii, I have lots of connected components. Every data point is its own connected component. As I keep increasing the radius, these will merge. And when I'm at R1, at that point, all of the data points are connected by these walls. So a lot of these features, a lot of these connected components are there immediately, and then they disappear very quickly. There's only one that stays forever, and that's the connected component that you see for R1 and R2, and that basically is there until R becomes infinity. The next thing that's more interesting in a specific example is the number of closed loops. And so you see that one closed loop appeared before the radius is equal to R1, and it stays all the way up to until the radius R2. So you see this square over here, it starts, it emerges when for a certain radius less than R1, and it definitely stays until the radius R2. And then there's a second loop here that emerges after the radius R1. So it emerges somewhere in this range, and it's definitely there when the radius is R2, and it disappears later as well. And so the way to think about sort of the robustness of these patterns is to think about the distance of these data points from the diagonal, because that means it essentially gives you an estimate for how long these patterns persist. So the further away you are from the diagonal, the more robust this pattern is, the longer it is present as you change the radius, and the more confidence you have that that really gives you a genuine feature of the data that's not just artificial.
Can I ask you a question here?
Yes, of course.
Oh,
yeah.
What are the two axes? I don't understand the X and Y axis.
Okay, right. So both axes are R. What I'm looking for is a certain, I check the radius of, the X-axis tells me when a certain feature emerges, and the Y-axis when it disappears. So for instance, if you look at this red circle, it's present when R is equal to R1. So it appeared somewhere between, and it's not present when R is equal to 0. So there's no circle here. These I cannot go from this data point to this without leaving my data set, while here I can go from ball to ball. So somewhere between the radius R is equal to 0 and the radius R is equal to 1, that circle appeared. So the X coordinate of that circle should be somewhere between 0 and R1. And it's still there when R is equal to R2. So it disappears at a radius R that's larger than R2. And so this square corresponds to this circle. It appears, so with the X coordinate, for some radius less than R1, and it disappears for some radius that is bigger than R2.
Okay, I'm having trouble understanding the big blue dot at the top. So
right.
I think that appears when everything is connected, but why would it ever disappear?
Correct. It would never disappear. So I think, right, good point. It would actually never disappear. This should really be at R equal to infinity. It's just sort of convention that people draw that, but you can think about that, it never disappears, right? Because once you have, as you said, if you make the balls bigger, at some point you have a big blob and it just continues to be a big blob. So it can never go away. So think about, that's a good point. Think about that blue dot here as being at R equal to infinity. So it never disappears actually.
Okay, and one final question, the two
Yes.
red squares. So how do you define a closed loop? Do they disappear when you have a triadic closure, or what's going on?
Right. So basically, that's a good question, and I didn't talk about this. So basically, the machinery behind this is that you think about these union of balls as simplicial complexes, and you basically compute the topology of those. And you can then identify the number of loops using the homology groups of that simplicial complex. So essentially the dimension of that homology group tells you how many loops generate your topology of that set that you're interested in. And so this allows you to extract information about how many independent circuits you have and also what the generators are. So you can basically, there's basically a Python code behind this that Ripster that was developed by different groups that allows you to take these data points, choose a radius, and then compute the topology, the homology groups. And that gives you exactly that information about how many loops you have, how many connected components you have, and this generalizes to arbitrary dimensions.
Okay, thank you.
Yeah, and I did not talk about that. Yeah.
These are persistence diagrams, right?
Correct. Yes, correct.
Thanks.
Yeah. So these are essentially, yeah. And so there's different ways in which you can present or visualize this information. And so these are persistent diagrams. That's one of the different ways. There's sort of barcodes, there's persistent images, there's very different ways, but I find it useful to think about when do these patterns emerge, when do they disappear. It gives you an idea for essentially the robustness underpinning these patterns. All right, good. So So essentially, we can use these ideas now to measure pattern features. We have these data points that are single cells, and I can use exactly the same process to understand number of stripes. I can understand spots, and I can do all of this by using this approach via TDA. The way that we're doing this is we're thinking about a fish image like shown in the left, this rectangle. We identify the left and the right endpoint so that we're on the cylinder. And so now every stripe becomes a closed loop. And so that allows us to identify, using this approach from before, to identify the number of loops and therefore the number of stripes. We are using, and then again, depending on when these, when connected components disappear, that tells us something about average cell-to-cell distances. So we can use that to determine those. And number of spots can also be done by looking at connected components over a certain region of radii. So we compute the stripe stripeness by using the arc length. So once we have identified a stripe, we can determine from the TDA analysis which cells belong to that stripe, and then we can essentially compute an arc length distance to determine the stripe straightness. The stripe width is essentially the distance between death and birth radii that tells us when these features emerge and when they disappear. The difference gives us essentially the width. And then for the spot size, we use single linkage hierarchical clustering. The roundness we measure by just essentially looking at PCA, principal component analysis, looking at the two eigenvalues and looking at their ratio. And then so just as an example, so we then, once we have identified a spot, we can calculate from the single linkage hierarchical clustering, we can understand or determine all of the cells that belong to it. And then, so here what you see is the spot center and the spot size and the roundness for two different spots. So this one is more round, and so you see that the roundness coefficient is closer to 1. 1 would be a perfect circle. Here we have something larger, and then we also have an idea for the average spot sizes. And so maybe I just give, maybe I skip this and talk just about the second one. And so we have a number of applications too that I wanted to talk about, but let me just focus on the second one, is model comparison. So what this allows us to do now is we can compare, in our case, different parameter sets for our model. So we use the original parameter set. We change in model B, we take our same model, but we change the length scale parameters that iridophores take to form, to change their form and transition from one form to the other one. And in model C, we really thought about removing one function that these cells have. So dense xanthophores and dense iridophores attract each other weakly. And so we remove that feature from the model. So you can think about this could be impacted by a certain gene. If you knock out this gene, that would have that effect. And so the question then is does this TDA analysis, does this allow us to distinguish these different models? And can we essentially do an analysis of determining whether the model output running through that TDA analysis tells us which pattern or which image belongs to which model? And so here it's sort of three typical model runs. And so you see that the wild type on the left is model A. You see that in model B, the black stripes are thicker, and there's also many breaks between or many connections between the black stripes. So there's breaks in the yellow stripes. And with model C, you see that the yellow stripes are much thicker and that the breaks actually occur in the black stripes. And so when we run on our model, and I'm just going to show you 3 different PDFs of the model simulations. One is dark stripe width, and then the yellow or light stripe width, and then the stripe straightness. And you see the first 2 really allow us to distinguish the different models. And so really these 3 sample images that I showed you on average are typical. So we run 1,000 simulations for each of these models, compared them, and you see that there is variability in the stripe width and also in the stripe straightness. But when you do a statistical test on those and this output, you can distinguish these 3 models using the statistics that we derived. So basically, that's a brief summary. So what that framework allows us to do, it allows us to sort of quantify patterns across scales. I talked only about one application. So we have a number of other applications and things that we plan to do with this to really apply this to what I said our ultimate goal was, namely to linking genotype and phenotype. And so one example is this application 3 that quantifies the impact of long-range signaling and the range of the signaling for melanoma birth on wild type and mutant patterns. And I think one of the big next steps would be to apply this entire approach to experimental images. And the main difficulty at the moment is that we need very high resolution images to do that. We have sort of, on the bottom is an analysis of a single image, just as a proof of concept, so it works. But at the moment, we have access to probably only 200 high resolution images. We are working with the lab of Christine Nüsslein-Vollhardt in Tübingen to get more images from them and apply this machinery to them. The next step would also be to look at more detailed model comparison. There's another model out now by Owen and collaborators, and it will be interesting to compare these two models. And then the last thing that's really, I think, is probably is exciting, but we haven't done anything with this yet, is that when Alexandria developed her model in the first place, the parameters were identified visually by hand. So really thinking about looking at experimental images of zebrafish, looking at the model output and trying to get them to coincide or agree as much as possible. And so with this more automated approach now, we could really do this in a much more systematic fashion, and that will be one of the next goals to do. So that was the end of the first part. So which took me actually longer than I thought, but this was the aspect of using essentially topological data analysis to quantify patterns in agent-based models of zebrafish patterns. The next thing that I would like to talk about is switching gears is detecting genetic architectures that are shared by different phenotypes. And so the goal was, in our case, looking at a range of diseases. We looked at 81 case-controlled phenotypes, different diseases. And what we wanted to see is, can we identify genes that are common among different groups of diseases? And the way that we did this is we used genome-wide association studies, and specifically the UK Biobank. So what they basically do is they have two pieces of information. They have typically do genome sequencing on individuals, not necessarily the entire genome, but a large part of it. And then at the same time, they link this with medical data. And in our case for the UK Biobank, that were ICD-10 codes, which are not really diagnoses, but they are billing codes for what kind of medication people get. And so there's a step in there where we set that the ICD-10 codes coincide or correspond one-to-one with a certain disease, and there are flaws with that generally, but that was the information we had. So we're going to we basically use that. And so the idea basically is that you have a number of cases, people who have a certain disease, you have controls. You look at changes in the genome at certain locations in certain chromosomes, on across chromosomes, and you're trying to see, is there a statistically relevant distinction between the mutations or the wild type and the mutation in the case that if you look at people who have disease versus people who are controls. And so the idea then is to see, can we collect this information and extract information about genes that are common or implicated in different diseases. And we're doing, as we said, we did this for the UK Biobank data. We focused on 81 case-controlled phenotypes, and we had roughly 350,000 European ancestry individuals in that data set that we used. So here's roughly sort of how the pipeline that we used. So as we said, we have information about the genome of these 350,000 individuals. And what we're looking at SNPs, so individual alleles and individual positions in specific genes, and look at what the different alleles look like. And so we can do this for all of the SNPs, all of these different single nucleotide peptides that are captured by the genome-wide association study. And then we use a tool that was developed by Suhini Ramachandran's lab, with whom we collaborated on this, which is called Pegasus, which takes these data and creates gene scores. So each SNP is within a gene, but there could be multiple SNPs within each gene. And what this code is doing, this approach is doing, is associating gene level p-values that allow us to see for each single disease whether that gene is relevant for that disease or not. And so what we eventually get is we get, for each of the 81 traits, we get a long row vector of the approximately 17,000 genes that we looked at. And these p-values range between 0 and 1. And what we would like to do now is to identify from these data whether we can cluster diseases together based on shared genetic architectures. The difficulty with Pegasus and the difficulty with this approach of going from SNPs to these gene scores is that you cannot just look at, let's say, the number of SNPs per genes that are different or equal to each other. There are lots of constraints. Many of these SNPs may actually mutate together. They may also recombine together. So there's many constraints where one has to be very careful in creating these gene scores and these p-values to not over or undercount mutations, because there might be correlations between those, and you have to be very careful that you take those correlations into account. So that's sort of the first spec. Is Pegasus is this algorithm that allows us to get these gene scores for each of the 81 traits. And then what the algorithm that we're using is Wings. That allows us then to do clustering analysis to identify prioritized clusters that corresponds to disease groups that share genes in a statistically relevant way based on that gene score matrix. And so Wing stands for word clustering to identify internal node branch length outliers using gene scores. This is something that was always new to me. So there's always this game of trying to come up with abbreviations. And so for us, it was basically getting Pegasus is Wings. And so that was the reason why we picked Wings for that. So let me go back and talk a bit about the clustering algorithm that we use. So it's really that last step. So we have this matrix, 81 rows, 17,600 columns. What we would like to do is we would like to find a way to cluster in such a way that we are confident that these clusters are relevant. And so if you just cluster with this, let's say you pick a certain number and you cluster these diseases into the number of groups that you specify, you don't really know how relevant these clusters are. And so what we wanted to use is we wanted to use hierarchical clustering that allows us to see how these clusters change as we change our clustering parameter, essentially. And so word clustering turns out to be great because it works well in high dimensional spaces. We're essentially in the 17,000 dimensional space. And it starts with single data points as clusters. So you have as many data points, in our case, 81, as you have as many clusters as you have data points. And then you iteratively merge pairs of clusters. And the way that you merge them is by one of these two equivalent formulas. So you can either think about, so M of C is the centroid, so the center of mass of cluster C. So you can either think about comparing the distance between the centroids of two clusters, weighted with the algebraic or the geometric mean of the size of their clusters. That's one way to think about that. The other way is to look at this formula that looks more complicated. What we're really trying to do is you're looking at the variance, the distance of points to the cluster. So you look at your centroid, you look at all of the different data points and you compute all of these distances, square them, sum them up. And what you want to do is you're doing this for your first cluster, you're doing this for the second cluster, that gives you these two quantities over here. And then you're doing this for the union of these two clusters. And what you're trying to do is you want to cluster Ci and Cj in the next step if that quantity, that distance is as small as possible. So if you change the variance between points in as small a way as possible. And so you just basically keep increasing this. Once you have merged two clusters, you look at the next set of clusters and you just keep merging until all of the clusters have merged and you just have the entire data point, the entire data set becoming together clustered in one single cluster. So what it really tries to do, it tries to minimize within cluster distances. And that is also the criterion for merging. So once you have that, you essentially get a dendrogram. So at each stage, you merge certain clusters. So you can think about organizing this in a dendrogram where you start with every single trait being a different cluster, and then you begin merging them step by step. And the next step is to identify relevant, prioritized, or significant clusters. And you're doing this by the branch length, which essentially tells you, is essentially the difference between when the cluster is created and when it merges. So, so the idea is that that quantity tells you essentially how tight this cluster is. So we look at the distance when it's created, distance when it ends, take that difference. The longer this is, in sense, we feel like the more meaningful that cluster is. And then we have a branch cutoff that is essentially given by looking at the overall branch length distribution and taking out the top 2/3 of that. And so what you see here is the outcome of Wings applied to the UK Biobank data set. And what we see is that there's one data point that's way up here that I didn't want to plot because everything else would be so small. So we identified several different clusters that we gave names based on the commonalities between the diseases in each group. So one is the metabolic, immunologic 1 and 2, polymetabolic, and then one cluster that is right at the border, which turned out to be actually meaningless. So there is definitely the potential for clustering diseases together that may not have enough genes that are shared across them. And so here's sort of not the full diagram, but the top level dendrogram that we have. So you see how these different diseases merge. The key is really this little table here that tells you how many genes are shared across the diseases in each of these groups. And so you see that 181 in the first one, 96 in the second one, 2 in the metabolic one, and really there's 0 in the assorted one. They share genes across pairs, but not across all of the members of that group. And then again with the branch length. So this allows us to basically sort out, come up with these groups based on the number on that algorithm that I described, and we also identify the shared genes, which could then hopefully be of interest to biologists to look further into what the relevance of these genes is and why they are commonly shared between these different diseases. So just a brief summary. So yes, go ahead.
Sorry, I just wanted to ask.
Yeah.
So in addition to the final output of this clustering algorithm, do you get any information from sort of the dendrogram itself and how long it takes before any of these clusters merge?
Yes, so we see, so the branch length here is essentially the information about how long it takes for them before, the distance between when they're created and when they're merged with a different one. So that's encoded in the branch length. The dendrograms themselves, I personally find them really misleading because it's very hard, because they don't really reflect the branch length. So you look at these pictures and I always think like, why is this not selected? Why is that not selected? And really is that the dendrogram is not a good way of capturing this information. It's really all in the branch length. The only issue is that in that area, most people think about dendrograms and not about branch length. So that's why we always show the dendrograms, but there's much less information in the dendrograms. I think what's most important about this is really which genes are implicated, which genes are underpinning the reasons for why they were clustered. And we can, afterwards, our analysis tells us what these genes are. And so here are sort of a few examples where we list a number of these genes. And we specifically list those that were already implicated specifically by medical studies or biomedical studies with specific diseases. And so we captured those plus others. And so then the hope is that since there are clearly genes that have been implicated already by by pathway analyses and other means, that hopefully that the new genes that we identified might at least be starting points for further studies. So that's the goal of why we are doing this. So we identified 8 prioritized clusters. 7 of those actually share significant genes that have been associated with at least one of these traits or diseases in previous studies. The exception is the assorted cluster. And the hope is that this provides candidates for drug design. And that's the long-term hope of this study. So that was the second project. The last one that I probably have to talk a bit faster about is aligning single cell data. And that's also the most recent one. And so we're just preparing that manuscript for publication. So hopefully actually at the end of this week is our goal for getting this finished. And so here's the idea. So the goal is to start with experimental data that measure, for instance, gene expression or methylation in single cells. And typically these are hundreds to thousands of cells where these data are, where these properties and these features are measured. And so you have, for instance, in the first set, you have cell 1 up to cell n, and you can measure the gene expression. You have a set of different cells, let's say 1 to m, but they're not identical to the cells used here, where you can measure RNA methylation. And so the question then is, then you get certain data points. You get data points for the gene expression data and data points for the DNA or RNA methylation. And you would like to align them to see what their relationship might be. So if you see cells with a certain gene expression level, can you relate that to DNA methylation data? And so how would that alignment work, and how would one do that? There's a large number of techniques known for doing this. Most of them make assumptions that you actually do have relationships, that you do know something about how these different modalities of measuring, how they're related, and also how the cells are related. So one common experimental strategy is to use co-assays where you actually can measure 2 different properties like gene expression and methylation from the same cell. And so then you know actually which cells these data points correspond to. So for instance, cell 2 here would be the same as cell 2 there. But these co-assays are limited to very specific techniques that actually allow you to work with co-assays. And so what we were trying to do is, can we come up with an alignment strategy that doesn't use correlations between different cells or experiments that were done on the same population of cells.
I just wanted to ask a clarification about this.
Mm-hmm.
So are the measurements of these 2 quantities, gene expression and RNA methylation, I understand you don't necessarily know whether or not that's from the same cell, but in principle, do you have each measurement for each cell? Or is this diagram on the left saying that there's some cells that you can measure RNA methylation for, others that you can measure gene expression for?
So right. So if you have co-assays, these 2 cell groups would be the same. So you can at the same time measure for one cell, you can measure multiple properties like gene expression and methylation. But for many other experiments, the cell groups are different. So you do not have the same. You do not have the 2 different data points for the same cells. So you might have half of the cells where you do gene expression, and the other half you do methylation.
And then, but the target is nevertheless to somehow correlate these 2 things at the single cell level. And then could you just explain the third column here on the right side? What are these 2 showing?
That would be just different. That would be, for instance, so these data points are typically it depends. So for instance, you could measure a number of different genes and the expression level of different genes. So you get whatever number of, let's say you measure 13 different genes and the expression level. So you get a 13-dimensional vector, and this is just a 2-dimensional representation of that output. So for each of the 2 methods that I expressed here, you would typically have different parts of the RNA sequence for which you would measure methylation. You would have different genes for which you look at the expression levels, and you would like to compare those full data sets with each other. In this case, what I show here are essentially two-dimensional representation, typically like PCA representations of these more higher dimensional data sets.
Thank you.
Right, and so if you know that how these data, how the cells or the features are related, then there's algorithms that allow you to do that. And so if we focus on this scenario, so we don't know that. We still assume that there is a shared common manifold structure behind this. Otherwise, there would be no hope of doing this. There's two algorithms that were developed in the last year or so that do that. So MMDMA and UnionCom, and they're doing this by constructing and embedding into common spaces. So essentially learning manifold structures. They have, both of them are minimization techniques based on neural nets. They have hyperparameters, 4 parameters in each of these cases that you have to set in order for this algorithm to work. And the main issue then is that you have to choose these 4 parameters, and choosing different parameters gives you different answers. So what we try to do is, first of all, minimize the number of parameters that our model has, and then also make the model or the approach scalable, which neither of these approaches can do. So they work for hundreds to a few thousands. When you go higher, they don't work anymore. And so here's the idea. So the way that we're trying to do this is using optimal transport. And so optimal transport, and I'm going to say in a second why we use optimal transport. So it's essentially trying to take like different sand piles and moving the sand particles from one sand pile to the next. And you want to do this in such a way that you minimize work. So you can imagine that you have this sand pile of blue particles, and you want to somehow map these blue particles onto the red particles in such a way that that you minimize work, and work meaning that if you take 2 particles in the blue sand pile, you move them over to the red particle, to the red particles, then the distances should be as close to each other as you can. So if you have a certain distance between the blue particles, you want to map them onto red particles that have approximately the same distance. So this means that you can sort of take a spade full of particles, move them over, and you don't have to worry about how you adjust the particles within your spade full of sand that you're choosing. And so here's an example for actually the optimal strategy for moving that double well probability distribution to this single well potential on the right-hand side. So you sort of, if you see that, so that matrix basically tells you which particles are mapped into which parts of the other sand pile. So what you're doing is you're basically taking this entire sand pile here, moving it to the left part of the red sand pile. You take this pile here and move it to the right pile. And that is in some sense the optimal strategy for doing that. And so more formally, what you do is you're trying to minimize this this quantity. And so what the key output is gamma. It's this matrix that basically tells you which particle is mapped onto which other particle. So which blue particle goes to which red particle. And the way it's doing this is not a one-to-one correspondence, but you're doing this with probability. So you're basically telling the output is essentially a matrix that said that this blue particle goes to this particle with like a 50% chance, to the other particle with a 50% chance. And so it really gives you a probability distribution of what the optimal coupling matrix is, but from a probabilistic viewpoint.
Can I ask just one quick question?
Yes.
Sorry, I think I missed it. Is the mapping DXIK in the upper right, is that going from blue to red, and then the other one going back from red to blue?
Right. Sorry, these are actually the distances. So what you're trying to do is you're going to try to map I to J and K to L. And what you're trying to do is you're trying to do this so that that distance between I and K is close to the distance between J and L. So you're trying to preserve the data structure, the sort of adjacency or closeness in the data sets. So the idea is essentially that what you're trying to do is you're trying to have, you have these data sets. The only intrinsic structure that you have is pairwise distances. That should mean something, because what you're doing is you have, I mean, these are the coordinates that measure like gene expression levels. And you would like in your mapping, you would like these to be preserved in as much as possible. So if two data points are close, so they have similar gene expression levels, you don't want them to map them into a demethylation to two DNA methylation data that are very far apart. You want them to be close as well, right? So close data points should map to close data points. And ideally, you want to do this for everybody, for all of the different data points that you have. And you can't, I mean, this is typically not possible. So you try to minimize the extent to which you cannot do that. And that's essentially what this does. So you look at you take any two pairs, you look at the distance of the 2 points in the blue sand pile, map them to the red, and you get that distance, and that gives you this term here. Then you look at the probability that actually I is mapped to J and K is mapped to L. That product then gives you sort of in a probabilistic sense, the amount of offset that or work or energy that you're creating. And then there's the regularization factor that you have to account, but essentially it's the first part that's the important part. So if, for instance, if the probability that I gets mapped to J is very small, then this term doesn't matter. But if you think that I should really be mapped to J and K should really be mapped to L, so gamma and the two gammas are close to 1, then you really want to enforce that these two points have the same, or these two pairs of points have the same distance.
I also have a question. And then I see there's also a question in the chat.
Sorry, I didn't have the chat open.
Also say that in just a moment. I was asking, so there's still some overall shift that might be there between the two data sets. Is that right? So you need somehow to have some absolute mapping from at least one data point to the other to get this overall shift.
So
I'm just imagining the difference, say, between some positive or negative correlation between the gene expression and RNA methylation. You could imagine that your data points would stay very close together, but how they map from one manifold to the other, perhaps with a reflection or a translation, would depend on the nature of this correlation.
Right. Yeah, that's a good point, right? So in this example here, for instance, there's two choices that you have. I could at the same time I could reflect the data points, right? If I say that half of the blue ones go to the left, the other half of the blue point goes to the right, I could switch this without changing this. And so so this minimization theme would not match that. It would not see that unless there's a significant distance. A significant difference between these two things. So if you have translation or rotation, this minimization technique would account for that. It wouldn't matter. But if there is two different ways in which you can obtain, in which you can minimize this quantity, then you would potentially get two, depending on where you start with your algorithm, you might get two different answers.
Right.
And so this is not part of that.
You could use actual, I mean, if you have a single measurement where you are able to get both of these things, you could use that to disambiguate.
Then you can use that. Correct. Yes. That's
okay. Right. Let me also make sure that I say Sasha Nelson's question. So he'd like to ask, does the relationship between the two domains mapped need to be monotonic? Does it need to be linear?
Yeah, it does not. So these could be manifolds in high dimensional spaces, and so really complex data sets. And for the Gromov-Wasserstein distance, it doesn't matter. So this process would work no matter what happens. So they do not have to be linear. They do not have to be monotonic. It would work either way. I mean, assuming that there is a commonality shared between those. So I'm running out of time. So let me, I'm already over. So let me just see. So essentially, just very briefly, let me just focus on this and go very quickly over this and not talk about the examples where we show that this actually works. So the idea of what we're doing is we're going to use this Gromov-Wasserstein distance. So what we're doing is we're starting with these data sets. We're looking at you have to create a distance. So this distance function is the key thing. And the way that we're actually using the distance is that we don't use the global distance in the data set, but we're looking at data points that are close to each other. So we are essentially doing a k nearest neighbor graph, creating that for a fixed value of k. That's one of the parameters we need to set. Then we can use that to create a distance. So we have now a network. We know who's connected to whom. And for each of these points, we can then look at, can generate a distance from that k nearest neighbor graph. Then we can, once we have that distance, we can apply the Gromov-Wasserstein distance. We get this coupling matrix, which is probabilistic. It doesn't tell us that cell 3 of the first data set gets mapped into cell 51 in the second one. It gives us these probabilities. And so what we then do is we use barocentric projection. So essentially the mean of all of the points weighted by the entries in the coupling matrix to see where each point would be mapped if you would really insist on mapping them. And that gives us the final matching. So in contrast to this previous algorithms, what we do is we have essentially only two different parameters. We have k, which actually is the algorithm is not very sensitive to. We have epsilon, which I didn't talk about, which is this regularization feature that is needed to allow us to actually make sure that the numerical algorithm converges. And so that is a parameter that does change the optimizer. And then the output is a matching, a one-to-one matching. But what we do is, in contrast to this previous algorithms, is we match one data set onto the second data set. We can reverse the order and do this the other way around. We do not take both of these data sets and map them into a common space. And with this, let me just go. So it works. So at least for the data sets where we apply this to data sets where we have co-assays, it works really well. And the idea now is in the future is to apply all of this to larger data sets and ideally think about some theory that allows us to explain why the alignment works so well. And with this, let me just thank my collaborators. So Alexandria Volkening and Melissa were involved in the zebrafish work. Melissa and Sam Smith, who is a graduate student of Sohini Ramachandran's here at Brown, worked on the genome, on the GWAS data set. And then the last project I talked about is together with Pina Demetki, Rebecca Santorella, who are both graduate students at Brown, Bill Noble, who's at Washington, and Rinda Barasingh, who's a computational biologist also here at Brown. And so I'm sorry that I went over time. I thank you very much for your attention. If you have any questions, feel free to ask.
Well, thank you very much for such an interesting talk. So feel free to ask any questions if you have.
I have a few questions. Thanks a lot for the talk. That was very rich, a lot of information. I had two questions. One was related to the first part. And my question regarding the first part, so the pattern formation is a zebrafish, was wondering if, I was wondering about the choice of having a cell-based model versus a PDE. And in fact, do you have a PDE that is associated to the stochastic system that you define? And in fact, you would have other types of quantifications for the pattern that you could use with a PDE. And is there a way to connect these two?
Right. So we decided on an agent-based model primarily because we started out with we really wanted to have a model where we can include all of the biological information that we had at the same time. So there were lots of information where they showed which cells repel each other, which cells attract each other, how fast they move. And the agent-based model was by far the simplest way to include all of the information and come up with a model that really describes individual cells and allows to make very specific comparisons between the experiments and the model output. So we also looked at, this is preliminary work. I think Alexandria is working with Jose Carrillo on this. What you can do is you can do a sort of a formal limit of that agent-based model and to try to see to come up to a continuum model. And what you get is not a PDE, but you get an integral differential equation. And the reason for that is really that you have these finite radii over which they sense each other, and you need to preserve these finite radii, otherwise the model doesn't work, or that limiting process doesn't work. And so you get integral differential equations in the end. And the main difficulty that we have, that we never really fully resolved, is the limit of all of these different interaction terms that are not clear to us how to generate those. So these cells move from one to each other, and that again is essentially taking averages over nearby cells and how many cells of different cell types are there. And so I think it's just going to be complicated to come up with a model that you can really obtain as a continuum limit. But I think you can just basically take those terms and then simplify them to come up with something that you could study. It's just, we just didn't get very far with that so far. But that's definitely something I think that is possible, I think. The question is how many assumptions you have to make on the way and how many simplifications you have to allow.
My question regarding the last part was about this the additional term that you had in the metric, because in the Monge-Kantorovich problem, or in the Wasserstein distance, the classical Wasserstein distance, you only have the first term, right?
Right.
The distance. And so what is this
Right.
A? Is it the entropy of the?
It's the entropy, essentially. It's like a diffusion term that allows you to basically make the gamma smoother and not discrete. And so the problem is with the original optimal transport problem is that typically you have to have the same number of points in both data sets when you map them. We don't have that here. And the other issue is that you really want to have, ideally, you would like to have a convex problem that you solve. And so that regularizer makes it convex. And then as epsilon gets smaller, your matrix gamma begins to become sort of less smooth and more concentrated. And so the number of minimizers can change as you decrease epsilon. And so the question is, what is the optimal epsilon that allows you to get a minimizer that you're confident about that minimizes this overall distance?
Okay.
But it's essentially like an entropy.
No more a distance, right? It's no more a distance.
It's right. Exactly. Yeah. It's a distance for the first one, but the second one destroys that. But it allows you to minimize that numerically, actually. Yeah, good. That was a good point.
I guess I had one quick question if there's time. Thanks. I was curious about the first part of your talk using this topological data analysis. So I'm curious if you tried any other sort of model-free methods of quantifying the patterns, like, I don't know, singular value decomposition and stuff like that, and how they compare, if there's any advantage to one or the other?
So we didn't do that. And so the main reason that we chose TDA is that we wanted something that really allows us to bridge the entire scale of patterns that we have, really from cell to cell to stripe distances, which are more macroscopic. And we wanted to not put in any specific information about average cell to cell distances or average stripe width into the approach. And what the TDA approach allows us to do is it has the scaling parameter R, this radii of these balls that you choose in it. So it really gives you patterns across scales. You can then see or look at what patterns persist over which scales and which ones are meaningful and which ones are noise. And so that's really why we picked that one. And we did not look at other methods because most of those were, most of the ones that we looked at didn't have that feature. And you would then have to prescribe the scales at which you want to look at before. And there's so much variability in these patterns that I think we were just worried that we miss out on features. And so that's why we did that.
Okay, thanks.
It's a very valid question. I mean, we just didn't, and that's the reason, for the reasons I just said.
Can I just ask one more question if it's not too much? I'm kind of wondering about how the mechanism that forms the patterns. So is it, in the first part of the talk, is it aggregation or some other properties of the kernels, like in those integral differential equations you were talking about?
So for the cell-based model, it's actually repulsion, and the repulsion, and then also, so there is essentially, it's essentially like a Turing or a generalized Turing mechanism. It's really like long-range inhibition, short-range activation, but the way in which the activation and inhibition is done is complicated. It's through enforcing cell death, for instance, of melanophores if there are too few melanophores around you. And so there's different rules that effectively give you like the short-range activation, long-range inhibition feature. But for each of them, it's a really robust system. You can knock out certain mechanism and you will always still see stripes. So there's redundancy in the system, but so there's different mechanisms for each of these two aspects of it that basically enforce that. And the overall outcome then is that you see the stripes. So it's very close to Turing on this kind of abstract level.
Interesting. Thanks.
And it's the same for the integral differential equation. It's the same features that would persist there as well.
Do we have any more questions for Bjorn? Alrighty, I guess we will wrap up here for today. I'd like to thank our speaker Bjorn again for such a wonderful talk, and also everyone for participating. So we will skip next week and rejoin in 2 weeks. Hope to see you guys then.
Thank you.
Thank you so much. Thank you.